All right, thank you very much to Nelson there for that very, very interesting pre-recorded talk. What I'd like to do now actually is we're waiting for another session to start. And it's going to take about 10 minutes, I guess. And while we're waiting, I thought it'd be really nice to go through the International Microorganism Day website, because this is really the best place to engage with us. And it's had a lot of work done on it over the past year. We've been contributing a lot. We've been upgrading and improving things. So I thought I'd just walk you through it a little bit. I mean, as you come to the first page, you see that we have the live stream here. That's obviously going to be important now for the next few days. So if you're ever wondering where to watch the live stream or catch up on what's going on, you just go to this link and you press play, and you'll be up to date with the latest in this marathon live stream. We've also got the program here as well. On the homepage, we've got links to our blogs, both our English blog, and we also have a Portuguese blog. International Microorganism Day was started in Portugal in 2017. And then we also have some of the featured blog posts and some more information about us and social media as well. So one of the things that we have highlighted a couple of times through the sessions already are the art. We've produced art for the blog, for the Women in Microbiology, but we've also produced art, or should I say you have produced art in the art contest. And you can see all of that in our gallery. All of it's online, and you're kind of free to use it, share it with attribution, and yeah, do whatever you want with them, basically. So we can maybe take a quick look here at the microbial art contest. So here you can see entries from 2021, 2022, yeah, they're currently being counted, of course, because we've only just announced the winner, but they will be there. And here are the entries from 2020 in all the different categories. And if we go back, you can see the pictures from past events on this page, but you can also see, as we mentioned earlier, the Women in Microbiology illustrations. And so these are in the gallery because these are all individual illustrations. You can use them on social media, download them, share them, do whatever you like with them. And they belong to a blog. And this is the other section that's had some work. In fact, I go to the blog last because there's one more thing before I want to go through the blog, and that's the educational resources. Last year, we introduced a Winogradsky column and a sourdough bread educational resource. There's a student's infographic and a teacher's pack. And this year we've added 2 more. So we've added making yogurt and making agar plates. So if you want to try them out, if you would like to do any of those, then just download them. They're freely available for you to go ahead and do that. And finally here we have the blog. So the blog, we've actually been trying to recruit bloggers and trying to get more people writing, which has been going well. You can see we've got contributed articles. If you're interested in contributing, you're more than welcome to send us an idea or a pitch or an article. If you're interested in learning about blogging or the format of the blog or skills that you need to start blogging, then we've also got a blogging bootcamp here, which I just scrolled past. And if you go to the post on the blogging bootcamp, you'll get all the information you need to actually join a free mini course where you can actually learn the fundamentals of blogging, and you'll learn basically how to produce a blog post for International Microorganism Day. So to do that, you can just go there and register your interest and you'll be enrolled in the course. And then you can actually, if you think you would like to write it, think it's something you'd be interested in doing, but you're not sure how, then that could be a very good way to start. You just do the course and then you kind of are taken through all the steps you need to write an article. So let's have a look. We've got some people in the waiting room now who I'm going to bring in. We're a little bit early, but that's okay. Let's see who we've got. We've got Devanita and we've got Saugata. Hello, Devanita. Hello, Saugata. How are you? Hello, hello.
Hi. Hi, Professor Dutta.
Welcome to the live stream. Yeah, I hope you're doing well. I'm going to hand over to you, Devanita, to host this this next session. I'll be here in the background for any technical help if you want it, but essentially now the floor is yours. So thank you very much, and I will take a back seat now and hand over to you.
Yeah, thank you, Joe. This session is on the Iron-Arsenic Microbiology, and since we know arsenic is quite a problem in many parts of the world, and iron plays a major role in the mobilization and especially the microbial action. So we'll hear about that from our two eminent speakers. They are global leaders in the field. And our first speaker, Professor Saugata Dutta. So right now we have him online. So currently he is a distinguished professor, Hammond distinguished professor at University of Texas, San Antonio. And earlier he was in Kansas State University. Professor Dutta has been working in this field for a very long time, along with the issues of water resources, water availability, and understanding the cycling of different metals and organic compounds. Today, with this, I would welcome Professor Dutta.
Sure. Thanks, Devanita. This is a real pleasure. Can you hear me properly?
Yeah, we can.
Okay, great. Yeah, it's a real pleasure to be here, and thanks for the invitation to be a part of this speaker series of this eminent FEMS, the idea that you chose is just excellent. So I am very happy to introduce our work a little bit to all of you, and also initiate some discussions maybe towards the end if time permits and stuff. So let me try to see if I can share my screen. Okay.
Yeah, we can see that.
Okay, can you share? Sorry, can you see my screen properly?
Yeah, yeah.
Okay, great. So good evening, good morning, good afternoon, wherever you are. I know we are at 2 different continents. Here it is, what, 8:30 in the evening. I had my real long day, and then I taught from 4 to 5:15, and then I prepared my talk for this evening that Devanita had invited us for. So I'll be talking about the role of natural organic matter, or NOM, in microbially mediated mobilization, and keeping the theme of iron and arsenic in mostly these reducing aquifers that we have seen all across or all over the world. Probably this is not just the couple of case studies and ideas that I will place in front of you is from the Bengal Delta, but this is not just the only ones that we know of. So hopefully I will draw some attention from you on that. Okay, so the outline of my presentation is such that it is divided into microbial presence or not. Let me just switch off my phone, sorry. It's microbial processes on Earth, microbial iron cycling, how natural organic matter comes into play, and then microbially mediated iron cycling and its impact on human health. I think this part will draw some attention, and then a few case studies from our group that actively we are working on. Okay, so we know that microbes are present everywhere, and this is something that this audience is fairly knowing about. On the ocean or on the subsurface or in the soils. We have not gone into the ocean from our research group, but definitely we have dabbled into subsurface and soils. And these are considered as the major habitats of the microbial life, right? And relatively smaller population of microbes actually can live in minor habitats, as for example, that's where we work mostly on groundwaters, wastewaters, even in animals or plants. So this is something that we already are knowing this to go forward with our idea of knowing the microbial cycling of iron and arsenic, for example. But here I just present the global iron cycle. Again, this is iron being a very important redox sensitive element in the environment, which microbes actually cycle through both assimilatory and dissimilatory metabolisms, right? Both of them act together. Most of the cases, the autotrophic microbes obtain their energy by oxidizing the reduced form of iron and sulfur, whereas the heterotrophic microbes, they obtain their energy by consuming the carbon in the environment, right? So in turn, they also oxidize the iron and sulfur. So cycling of iron is very important in geochemistry, especially with this soil sediments, groundwater geochemistry, and is incomplete without really studying the roles of microbes. So knowing that microbially iron is cycled, microbes love the redox process, right? So as there is always a transfer of electron, and they obtain the energy for their survival from this electron transfer. Now, the chart to the right actually indicates that various mechanisms by which iron is cycled in the natural environment through the microbial oxidation or reduction reactions. So this is something that is important to keep in mind when we have that process of exchange of one electron that provides the energy to the microbes. Okay, natural organic matter. So the natural organic matter, as we all know, can be really divided into various fractions, right? As shown in the figure to the left. And this is based on the size of the molecules, the elemental groups or the functional groups, mostly composition. For example, the biggest cycle, the biggest circle really is the total organic matter, right? I mean, total organic matter. And then we have the larger cycle of the DOM. Now, for example, a lot of the dissolved organic matter contains the dissolved organic carbon, right? The nitrogen, the phosphorus, which is based on elemental composition. Now, the DOM is also classified as either humic or non-humic substances, which also play an important role in the mobility of these metals or metalloids. In this case, one is iron for sure. Okay, so this picture actually shows the typical sources of, so we say that, release of organic matter from natural water. So as you can see, there are terrestrial sources such as, higher plants, aquatic sources, such as the microbes or the algae. And then figure to the right, to a little bit bottom, shows the various processes and transformations that this dissolved organic matter undergoes before we characterize it in a sample. Now, these processes could be either photochemical and chemical transformation, as well as it can be microbial processing. Now, while characterizing this dissolved organic matter in water samples, it is actually important for us, these results in the context of field results also, to compare them with. And that's what we do in most of our cases. So the figure this here, I'm hoping that this is very familiar to the audience here. It's kind of the size-based classification of the organic matter. Nominal size is that 0.45 micron is used to distinguish between particulate or the dissolved organic matter. But some studies have shown that the 0.7 micron or the 0.2 or the 0.1 micron filter sizes as well. So therefore, the distinction between the particulates and the dissolved organic matter is often, I would always consider, as operationally defined, right? And so you can see that the molecular weight of the various dissolved organic matter components are here, right? So bacteria and clay, for example, or clay humic metal complexes are sometimes particulate, and sometimes they are dissolved. Now, this classification can help in the DOM characterization that we do most of the times. Okay, so one of the research areas that my group are currently doing is working on microbially mediated elemental cycling and its influence on human health, or impact on human health. As we know that water quality is directly linked with human health, many microbial processes after the quality of the life by dissolving elements that are harmful to humans. And one of them we know of is arsenic, and I think you will hear a little bit about it after my talk also. So arsenic is a naturally occurring metalloid, and it's a carcinogenic too. It's considered carcinogenic, a class 1. And it occurs in soils, sediments, and they are mostly of geogenic origin. It enters the water systems by the sediment-water interactions and the microbial processes. On the top right, we show the Bengal Basin. That is, most of us have worked there. Major rivers of the Ganges, Meghna, and Brahmaputra is known to be arsenic affected. And the microbial processes here play an important role in mobilizing arsenic, which my group is mostly studying. But I would like to share some of the findings from there. Okay, so this is a very quick overview of some of the work that we have been doing in our group, mainly to understand the hydrologic, the geochemical, and the microbial controls of the mobilization of arsenic in groundwater. And we actually use all three, field, laboratory, and modeling-based approach to conduct these studies. Okay, so this schematic actually represents the biogeochemical processes that happens in a shallow reducing aquifer in the Bengal Basin, right? And in this case, the labile organic matter from the aquifer sediments serves as a food, as an electron donor essentially, for iron-reducing bacteria. Now, older or recalcitrant organic matter in sediments is relatively less labile for microbes to use as their food. Now, therefore, it was hypothesized that such humic-like organic matter can accelerate the acetotrophic iron reduction process via this electron shuttling. Aqueous complexation is there also, and then competitive sorption. That's also, other elemental ions like phosphates quite very easily demonstrate. So the microbial processes that are in the groundwater that follow a thermodynamic ladder, in this case, that's what is shown on the top figure over here. And these heterotrophic microbes can use the organic carbon as a source of energy, just like we eat and breathe, right? I mean, these microbes eat organic carbon and breathe not only the oxygen, but a variety of electron acceptors. For example, the oxygen or the nitrates or the manganese, iron, sulfur, and hydrogen, and in a sequence, like as it is shown here. So for example, oxygen is most favored electron acceptor for the microbes, whereas hydrogen is the least, right? And others are kind of falling in between. Okay, so having said that, we can see from the figure that as the oxygen is depleted in the system, then the system also becomes anaerobic, right? And in the Bengal Basin aquifers, we have seen that the sediments that are rich in iron-3 minerals, arsenic also often adsorbed on these minerals. And that's where the arsenic relation comes in. When the microbes reduce the iron minerals and dissolve them in the groundwater, arsenic is also followed, meaning they are mobilized into the groundwater. So the supply of organic carbon into the aquifer actually dictates how much arsenic will be mobilized or solubilized as it supports the microbial life, right? So this schematic actually here at the bottom shows that various mechanisms rather by which microbes can mobilize arsenic in the groundwater that can use organic carbon as a food. Okay, so having said that, we have also looked at the microbial communities that are present in high arsenic aquifers in Bangladesh. This is a work from Bangladesh, strictly in MATLAB. In the picture, you can see that the sediments were collected from the aquifer, and then they were analyzed with evidence of the microbial presence communities. And we do observe that some of the bacteria groups are very well known to reduce iron-3. Laboratory experiments were also done with various strains of the iron reducing bacteria in presence of different types of organic matter, such as either like the acetates or recalcitrant organics like fulvic acids, to inform how the microbes can utilize the available organic matter. Okay. So over the last decade, several studies have been done that our group have done regarding the groundwater samples from arsenic contaminated aquifers from all the way from West Bengal, India to Bangladesh, and found that in general, the arsenic concentration correlates with the dissolved organic carbon concentration. However, there is still a heterogeneity. That means that some high DOC samples contain very low arsenic and vice versa. Now, the question that comes or remains that, how can we explain this type of heterogeneity using the bulk DOC concentrations in samples? We hypothesize that to do this study, this is due to the reactivity of the recalcitrant the dissolved organic matter in the aquifer. So we'll try to prove that in our subsequent few slides. So there are several techniques that is used. Again, this is quickly showing that we use the absorbance and the 3D fluorescence spectroscopy to characterize the fluorescent organic matter in the water samples, and various functional groups we look at, but definitely the different field samples are analyzed with these. So here are the results from the field, right? So we found that iron bearing high arsenic groundwater, or iron bearing high arsenic contaminated, much higher proportion of terrestrially derived humic-like organic matter, which is likely to be involved in this electron shuttling and other processes that accelerate the microbial iron and arsenic mobilization. Okay, so having said that, the microbes sometimes use some smart ideas to obtain the energy, right, from either recalcitrant organic matter like humic or fulvic acids. And these humic substances are not very good food for the microbes to start with, and they are considered non-labile. So these humic substances can form the aromatic rings and can be used by the microbes as an electron shuttle. So this is what is shown in the schematic over here, and we can delve with that a little bit more later if wanted. But let's go to the crux of the matter. So, however, there was no this is experimental evidence to show that there was no direct evidence that the Bengal Basin aquifers contain the humic material that is actually involved in the electron shuttling process. So in this part of the study, we actually performed a laboratory experiment using humic substances to isolate from the high arsenic groundwater in Bangladesh and use a geobacter as iron reducing bacterium to calculate the electron accepting capacity of the Bangladesh groundwater humic substances. Okay, so these graphs actually are shown to summarize the results from our work from a part of the Bengal basin other than Bangladesh. It's also from West Bengal on the arsenic mobilization. And we observed that relatively younger Holocene aquifers contain the higher concentrations of dissolved arsenic as shown in the red symbols over there on the map, I mean on the plots. And also you can see these concentrations of iron, manganese, and dissolved organic carbon that are also elevated in some samples with high arsenic. Now, this is because iron, arsenic, and manganese are all released during this microbially mediated reductive dissolution process, which is fueled by the dissolved organic carbon. So the concentrations of some of the other co-occurring elements, like what I show on the plots to the right, actually is of concern, or some are not of concern right now, but should be monitored in the future. The graph that I show on the left bottom over here, that actually shows the close relationship between the dissolved arsenic and the dissolved organic carbon at multiple sites in Bangladesh. I mean, also mostly in Bengal basin altogether that we have studied so far. Okay, so let's go into the another ongoing study that we are doing. It's about the dynamic iron curtain, or also we call the natural reactive barrier, right? Now, these dynamic iron curtains are usually at the boundaries between the aquifer and the river, right? So you have the oxidizing river water and the reducing groundwater. And what I show here are a dry season, monsoon season, variation of this barrier, so to say, the expansion of the barrier and then contraction of the barrier also. And then at the same time, we show that how these river waters are either in the form of groundwater discharging from the river to the actual groundwater rather, and upwelling the groundwater, or the other way around. So this type of natural reactive barrier is actually formed of mostly the high iron content, and so iron oxyhydroxides, goethite, even limonite, or sometimes iron oxyhydroxides are the main component out here. It's very difficult to analyze it. We had a paper a few years back talking about from the Meghna River, and we are currently doing a work funded by the NSF to look at that more critically, this presence of this natural reactive barrier. Okay, so what we did was we collected the pristine sediment cores from the Meghna River and also from the adjacent shallow aquifer with high arsenic groundwater. The locations of the cores are shown on the map on the top, and also in reference to the Meghna River, right? And the depth to the water table is given along the different sections of the cores that we are showing. So what did we do with them? Basically, these collected sediments were performed as a leaching experiment to mimic the mixing of the fresh river water into the aquifer. And these extracts of the sediments in ultra pure water after the experiments were done were analyzed for variety of inorganic and organic parameters. And I will try to kind of illustrate that quickly. So the results actually indicate that the sedimentary iron and the arsenic and two other redox sensitive elements like cobalt and vanadium in these river banks. Now you can see that about a 40-meter depth interval, or rather 40-meter distance from the river, we observed that these enrichment of the iron, which could be seasonal natural reactive barrier. So also we note that the enrichment of the arsenic and other redox sensitive elements slightly varied from the location of the iron. So not that all the places where the iron was high, arsenic was also high. So which may be attributed to the differential oxidation of iron and arsenic upon mixing of the fresh river water, right? That's more oxidized river water. Okay, so this is also another way of looking at it. It's more like of a colored diagrams to understand that where from the river that we see these type of iron enrichment, and as such what the iron enrichment is correlated with, as I was mentioning about that vanadium, cobalt, and also mostly iron and arsenic. So we also looked at the in this case, these are looked at the loss on ignition, and we show that there is a proxy, or loss on ignition is used as a proxy for the total organic matter content of the sediment samples that are found with some silts and clays obtain or contain the highest amounts of the LOI. Now, while the riverbank and aquifer sediments contain much lower in this case. Now, interestingly, we observed that the leaching experiment, that the riverbank aquifer sediments, although they contain the similar bulk organic matter, their water leachability was drastically different. For example, the aquifer sands leached much more organic carbon than the river sands. Now, we also should note that even though the silts and the clays, like in this case, the silts and the clays to the right-hand side, you see that as the silts and the clays are plotted over here, where the water leachable organic carbon content, even though the silts and the contain much higher organic matter content, they did not leach that much of organic matter during the experiment. So we did the fluorescent signatures of the organic matter that leached during this experiment that actually varied significantly between the riverbank sands, the silt, and the clays, right? So the silts and the clays in this case contained humic-like and terrestrially derived organic matter, whereas the riverbank sands, they contained the fresh, relatively labile organic matter, and fresh aquifer sands contain a mix of the two. Okay, we shouldn't say fresh, but at least to us, when we collected them, when we analyzed them, they were pretty fresh. Okay, so this is just a cartoon to show that these results actually suggest that the arsenic enrichment was present in the Meghna riverbank. Now, the cycling of the arsenic and iron in the riverbank appears that to be different in this case than the typical like shallow aquifers in the region, right? And this is an ongoing study. We are currently pursuing more additional analysis, including sequential extraction of these sediments. Some synchrotron analysis is going on, and also quantifying this natural organic matter and this iron arsenic complexes in the sample as much as we can. So this is where we would like to have a summary of our findings so far. Surely we see that the microbes do play a vital role in the cycling of the redox sensitive elements, in this case iron and arsenic, which in this case arsenic at least is an important human health factor, and to some extent iron too. And then we did confirm that the microbial cycling of the iron is influenced by the presence and characteristics of the natural organic matter, right? And the natural organic matter that reacts with the arsenic and iron in several ways, as I said, aqueous complexation or competitive sorption, or even with the microbes via the electron shuttling. So in the riverbanks of mostly the seasonally fluctuating rivers, like I showed in case of Meghna, partly Ganges is like that too. And these are all meandering rivers. The presence of the iron cartons are quite a bit, I would say, interesting to point out. And they actually show a strong influence on the hydrology that controls the iron and arsenic cycling. All these caps that you saw as the barriers, these are enriched in iron minerals. And we see the mostly the arsenic getting absorbed on this iron-rich barrier, if you may call in between this aquifer sediments and the river. So the microbial cycling of iron perhaps influence the mobility of other oxyanion forming trace elements in addition to arsenic, like we showed for vanadium and cobalt, but definitely others need to be paid more attention also. Therefore, it is of prime importance in a human health perspective, again, not just for iron, but for the other elements that we just talked about. So with this, I will be happy to take any questions or any discussions to follow. Thank you.
Thank you, Professor Dutta. That was a very interesting talk, and definitely very interesting for me. I work on a similar line. I have pretty much a lot of questions, but I would also like to ask the host if we have any live questions from the audience.
We don't have any live questions yet. Sorry, we don't have any live questions yet, but I'll jump in if I get any. I'll either write them to you in the chat or I'll open the microphone up. But go ahead with your questions.
Okay, thank you. Thank you, Ben. So I have one question, like to start with the humics in the deep aquifer. So because in one of your slides I was looking at, and you said that there is an increase, and then there was a shoot up after 30 meters or so. And that seemed pretty interesting, like what is happening in the deep aquifers, because then we can think of when we talk about the safe and the unsafe aquifers, and where with some places where the microbes are in action and some places where they are not, and that's why they some of the aquifers, the Pleistocene are safer. So why are you seeing these sudden increase in the deeper aquifers, or are these humics? Or
yeah, probably you are definitely I would say that it's on the right track of thinking that probably the deeper aquifers may have a higher quantity of the fresher humics. Now, how do they retain back at that depth? After a reasonable time frame, like for example. But if you can really imagine that these systems are so much fluctuating with time, meaning that they are so dynamic environments that probably much more of this upheaval of the sediments, especially with these riverbanks and the aquifers together, with the course of changes of the rivers, and these are, as you know, probably many of these stalwarts in this direction have pointed out that these are the paleo-oxbow lakes or the paleo channels, right? So definitely these are more of the reacted sediments that way. So when we talk about these more reactive sediments and they are going into the deeper aquifers, I think that characteristic do change. And probably the addition of fresh organics also, like in this kind of terrains, are quite often very possible. And now whether they percolate down to those depths is a big question. I do not know. But I do know, we do see that there are a lot of impermeable caps, for example, like the clays in between. Now, if these clays are also playing the same or similar action like the organics is another thing to understand. So one of the parts that we did not pay attention to yet, but we love to, is this clay, NOM, and then this arsenic or iron kind of the complexation to understand. The organic carbon that is actually more hosted in the clays rather than organic carbon itself within the other mineralized parts of the sediments. So yeah, with that in mind, I think that's I think a lot more work can be done on this direction. Very good question.
Yeah, so this was a very interesting talk, and something that really caught my mind was the dynamic iron curtain. So yeah, this is really interesting what you're seeing in the riverbeds, and I would definitely like to explore more. I mean, right after just this talk, I think I would start reading on that. So do you see methanogenesis in
Yeah, not to that extent of a complete methanogenesis. No, it's not a complete, but we do see signs of methanogenesis do start to happen in some of these sediments. One of the things that we did do is we haven't been able to analyze them yet because of all the pandemic upheaval. Basically to date some of these sediments and then see that if which dates of sediments, and these are very freshly collected, right? So probably with the recent river upheaval or the floods that happened in the couple of years back, the few centimeters of the sediment has been trapped in that within that time frame, right? So I would say that in our geologic time span and the sediments, I mean, these are fairly recent sediments, right? But the proportion of the organic carbon is different. So we kind of diversified that to see that how much organic carbon or type of organic carbon. And then now we are going to date these sediments to see that, if these are really, and probably the C13 also will help us to understand, and also C14, that whether there is any correlation of this freshness of the sediment, right? And whether they are whether really this reactivation of these sediments can lead to so-called making the sediments old enough to either retain or make the organics inactive. Now, I do not completely believe that it's a case, but I have seen in papers that they are analyzing. I mean, they are postulating for, like our natural reactive barrier work. I mean, there are multiple of work that has been done in other countries and other riverbanks, and they are seeing the same thing. The only difference probably is the proximity to the river. Sometimes, like we did pretty much a transect from the river to the aquifer, like a 80 meter, almost an 80 to 85 meter transect we did from the actual riverbank to the aquifer, right? So we do see a very prominent sediment characteristic, and along with that organic matter characteristic varying. When we had to sample the river, the river bottom sediments, I mean, the organic carbon basically is, I mean, it's full of organic carbon, let's put in that way, right? And these are heavily populated areas, so all things to be taken considered, right? But these are still on the so-called the river banks. We studied only the Meghna in this study, but we did also study with my Columbia colleagues up in the Brahmaputra, not on the very source area, but more towards the delta. And we did see much less of this iron barrier being forming. Meghna is critically important in this. I have not seen much studies on the Bhagirathi or the Ganges that way. But surely if we do study in kind of pristine areas along those rivers, we will find the same. That's our kind of aim of the future studies that we will try to take.
Right. Great. Thank you. Thank you so much, Professor Dutta. We have our next speaker in line, but this was a very interesting talk, and thank you for delivering this.
Can I stay back for the next talk?
Yeah, sure, of course.
Okay. Thank you very much.
Hello, Professor Sar. Good morning. You are muted. I think you have to unmute yourself.
Yes. Can you hear me now?
Yeah, we can hear you.
Okay, so let me share my screen, and please, shall I go ahead?
Yeah, I will. So I will introduce our next speaker to the audience. Our next speaker, Professor Pinaki Sar, and he's from Indian Institute of Technology, Kharagpur. And he is in Kharagpur since 2004. And before that, he was the Boys Cast Fellow at Rice University. And then he also worked in some other institutes in India. And he's also one of the leading arsenic iron microbiologist globally. And he has been working in this field for a pretty long time. So I have been following his work for, I don't know, since I started my PhD. So with that, I would like to welcome you, Professor Sar, on the International Microorganisms Day at FEMS. So,
thank you, Dr. Ghosh, for your nice introduction, and it is indeed a matter of great privilege to talk about the arsenic microbiology in this International Day of Microbiology. So I'm going to share my screen. So please let me know that if you are able to see my screen. Okay. Are you able to see my screen?
not yet.
Okay, so because we are not using this particular Zoom platform for our regular classes, so you have to just train me possibly. So should I go to the desktop, or exactly where I have to go? So Microsoft PowerPoint.
So once you have opened the PowerPoint, and then you have to click the share screen on the bottom of this Zoom screen.
Mm-hmm.
And there's a green symbol on the
green thing is coming, but
yeah. And okay, once again, Ben, Professor Sar has access to share his screen.
Yes, he does.
Thank you.
So at the bottom of the window, yeah, just click on share screen and it should give you a choice of options which window you actually want to share, and then you should choose the PowerPoint.
Well, so when I go for this share screen, a couple of options appears: desktop one, whiteboard, iPhone, Google Chrome, Microsoft PowerPoint.
Choose the PowerPoint one.
Okay, PowerPoint, and then share. Then another dialog box is coming: allow Zoom to share your screen, cancel and open system preference. Okay, so maybe security and privacy to grant access. So I don't know. I'm sorry. How to go about this security? Ben, can you please quickly help me?
So you'll have to allow access. So when it gives you any options to do that, you need to allow access for Zoom to share the screen. So if you accept that, it should let you.
Okay. So how do I give this access actually?
Is there an allow button somewhere?
No.
Maybe you can share your desktop
Okay, let me try.
and open the.
Files and folders. So, is it visible now by any chance? I don't know.
No, not yet.
It's not coming.
Can you share your desktop and then open your PowerPoint on the desktop and minimize the screen?
Yes, it is already open. It is already open.
Okay.
It is already open. I'm just looking at the security. I think my Mac machine is not allowing me because it has some security issues, file and folder. So I'm really sorry about this. So let me just quickly.
No problem. If you want, you can send it to me and.
Okay. Shall I do that?
Yeah.
Yeah. So I don't know.
So we're going into a break after this session, so we can go over by 5 minutes or something like that, so we can get the time back. That's no problem.
Okay.
The file is very big, so I have to go by Google Drive. So I think then, Devanita, you can go, as Ben suggested, go for a quick break, and then I think by this time we'll be all set.
Okay.
No,
okay.
sorry, I was saying instead of going for a break now, we do after, yeah, because it will only be a short break now, so there's not much point sending people away and then coming back, but we can overrun a little bit. But what I suggest we do, yeah, we kind of do the transfer of the file now. And as soon as you've done the transfer, Dr. Pinaki Sar, maybe you can give a little bit of your introduction while Devanita is downloading the file and kind of setting it up, so we can just hear a little bit about the intro of your research, and then when you're showing the figures and the slides and stuff, then the presentation will be ready to go. Does that sound okay?
Yeah, I'm okay with that. So I'm just sending through, uploading in the drive, and maybe in a few minutes, Devanita will get it.
Brilliant.
I'm really sorry for this mess up,
problem.
but
Seeing this all the time
I'm not,
these days.
yeah. So it's taking time. The file is little big.
No problem.
Devanita, for a while, I'll just keeping myself mute and I'll try if I can do something.
So, Devanita, maybe you can tell us a little bit about your work and your research in the meantime. I mean, yeah, you're the host, but we don't get very much opportunity to hear from the host always. So that would be nice.
Right. Yeah, definitely. I will take this opportunity to talk about something I do. So I work very closely on what Professor Dutta is working on. Working on the organics and how the organics are affecting the iron-arsenic microbiology. So it's the biogeochemistry of, it's a very regional study, but my current work is focusing more on the arsenic iron microbial role, and then how iron specifically affects the co-precipitation of arsenic. Now I'm using the ideas into a mitigation method, say filters to clean up water. But earlier I have worked on the role of organics, say larger molecules, alkenes, alkanols, and then these are not very labile or very dynamic, but what Professor Dutta is looking at, the smaller molecules, humics, these are more dynamic and then easily they break down and more actively participate in the microbial metabolism. Whereas the larger molecules, they are more recalcitrant, they stay in an environment for a pretty long time, and then they are also not degraded by the common microbes. So they kind of tend to impose a natural selection in the environment, and then the microbes who are able to grow and utilize these kind of the recalcitrant molecules, they kind of tend to get naturally selected, and they are more abundant, and then they tend to control the environment and the fluxes of different elements. So that's the idea that I am working on, and for pretty much a longer time, say 10 years. Yeah, so right now in Delft, the work is more on the filtration based on the iron arsenic participation and co-precipitation. So yeah, so Ben, you have been hosting this since yesterday. I am like joining in and out on YouTube for like, I don't know, and I was like thinking that these two guys, Ben and Joe, must be very tired at the time, if you will, because Joe, I know, he's in Netherlands too. So
I'm actually in Sweden, so we're quite close. Okay. We're not as far away as we are, but and we're on the same time zone as well, which is useful. But yeah, it's been a long day. Yeah, it's kind of tiring, but yeah, we're doing this marathon, you know, so there's got to be a little bit.
This is early in the morning, I think. What time is it? 4?
For me now, it is 4:15. Yeah.
Right. Yeah.
Yeah, so it's early.
Yeah.
But yeah, I did this last year as well, so I'm kind of used to it. So it's not such a big problem.
Okay.
Oh, what's this we see?
Yeah.
Oh, a presentation.
Okay, so we have Professor Sar's presentation here, and yeah, maybe Professor, you can go ahead with this. Thank you.
Are you able to see the presentation now?
Yeah, we are able to see, and maybe you can put it in a full screen mode, and yeah.
Sorry, Ben and everybody else for this mess up, but you understand that this is not intentional at all, and I am not used to this Zoom platform. I'm really sorry for this.
Of course, it's no problem. No problem.
Okay, so shall I move on and start?
Please.
Yeah. So today I'm going to talk on microbial world of arsenic contaminated aquifer within the Bengal basin, particularly our study area is West Bengal, India. And as you possibly know, that elevated groundwater arsenic is a global threat. And it is considered to be the largest poisoning of a population in history, with very large number of populations are under threat, like more than 200 million people, through drinking the arsenic contaminated water or ingestion of the arsenic contaminated food. And arsenic is also having very, very severe health implications. So now, if we look at the contaminated areas, the groundwater contaminated areas in particular around the world, we see that the South Asian and South American regions are the worst affected. And if we look at particularly the South Asian countries, it is estimated that over 190 million people are currently affected by arsenic contamination in the groundwater. And these contaminated aquifers are mostly hosted by sedimentary basins and close to the modern mountain belts and deltatic regions. And to be more precise, over the Southeast Asian contaminated regions, which are hosted by young alluvial aquifers of Himalayan origin, what we see that the rivers which are continuously feeding these alluvial landscapes in and around the places of Bangladesh, India, and China, and all the other areas, including Pakistan and other places. So they have contaminated the underground system. And in particular, with reference to India, what we see that almost like, except a couple of states, it is arsenic contamination in groundwater is now detected in almost every corner of the state of the country, India. Now, the major questions of geobiological significance remain: what is the mechanism of arsenic mobilization in the aqueous phase? And what are the factors which control this aqueous arsenic level? Now, these are two very fundamental questions, and perhaps it is now going to be 2 decades that scientists are trying to answer, microbiologists, geomicrobiologists are trying to answer these simple questions. And yet these questions are not completely answered, maybe because of the heterogeneity, because of the sampling issues, and many other parameters. And what is important here to understand is that these answers to these questions will allow us to gain a deeper insights into the relationship between the subsurface microbial life and the physicochemical environment of the alluvial aquifers. Now, if we look at the source of this arsenic, this is basically a geogenic arsenic. It is basically derived from the rocks which are originated, or the minerals originated from the Himalaya, and they are deposited in the alluvial landscape within different arsenic-rich iron, manganese, aluminium oxide minerals, and they are hydroxides. Now, if we look at this simple cartoon, we'll be able to understand that the presence of arsenic within these iron oxide hydroxide minerals is a very common phenomenon within this alluvial landscape. And arsenic remains mostly as arsenic 5 arsenate ion adsorbed onto these mineral phases. Now, over the years, what we have also learned that the microorganisms, they play a very critical role in transforming this arsenic, which is bound to iron, either directly by transforming arsenic itself, or by transforming the iron molecule present within it. Now, it is considered to be significantly contributed by microorganisms, and the indigenous bacteria, they transform this arsenic basically through redox transformation, which allows the alteration of the arsenic speciation. And arsenic 5, that is the arsenate, which adsorbs strongly on the mineral surfaces, gets into the aqueous phase as soon as it is reduced because of the microbial activities. Now, when we look at this particular process, though originally the arsenic is from these volcanic rocks and other through the weathering and erosions, it is now a part of the alluvial landscape. And this arsenic, which is deposited at within the secondary source, we called, because it is not the primary source of arsenic. However, this arsenic which is present at the secondary source is continuously being transformed by the microorganisms who are present in these alluvial aquifers. And these transformations, both the oxidative and reductive transformations, they carry out the relevant reactions which facilitate the release of arsenic under the conducive biogeochemical conditions. So the cartoon itself explains that these microorganisms of diverse types, they colonize, they naturally live within these arsenic hosting minerals. And these process of arsenic we call geocycling by these microorganisms, they are highly constrained by chemical and physical factors. And this could be one of the reasons that the complete complete picture of the arsenic geomicrobiology, or the role of microbes in arsenic contaminated aquifer, iron arsenic transformation, is not completely elucidated because of the spatial and temporal variations which we encounter in these aquifer regions. Now, what has been clearly established that there is a complex interplay of geomicrobiological processes in arsenic mobilization. So here you can see that the iron oxide hydroxide minerals, they host the arsenic mostly as arsenic 5, and primarily the iron reducing bacteria, which are also sometimes called as IRB, they reduce this iron as a terminal electron acceptor and allowing the arsenate to be mobilized or dislodged into the aqueous phase. These arsenic 5 is subsequently reduced by bacteria who are called arsenic reducing bacteria, because these bacteria, they contain a particular enzyme which is called cytosolic arsenic reductase that allow these bacteria to reduce arsenic 5 to arsenic 3. On the other hand, there are dissimilatory arsenic reducing prokaryotes which can directly reduce the arsenic 5 as a part of their respiratory metabolism and lead to the release of arsenic 3 into aqueous phase. In both cases, the arsenic 3 is released into the aqueous phase, and possibly that is one of the reasons that the aqueous arsenic is coming through the microbial activities. This is pretty well known for past, at least past decades. But here is another process which actually takes the process in the reverse, like arsenic oxidizing bacteria. They utilize arsenic 3 plus, they oxidize arsenic 3 and converts this arsenic 3 to arsenic 5, and arsenic 5 get adsorbed onto the mineral surface again. So essentially, this is a kind of very intricate and complex process where both iron and arsenic reducing bacteria, iron and arsenic oxidizing bacteria play a role or play their roles in the heterogeneous aquifer environment. However, this kind of reductive dissolution that is actually shown in this cartoon, it necessitates the absence of oxygen, because if oxygen is there, possibly iron and arsenic is not going to be reduced, at least in the dissimilatory process. Supply of organic carbon is required because thermodynamically otherwise the process will not be feasible. And of course, we must have appropriate biocatalytic bacteria who can, especially bacteria, because role of archaea is yet to be clearly revealed in this kind of aquifer environment. So the presence of these appropriate bacterial strains are also very much important. So we initiated this piece of study, this particular part of the study, in order to investigate the role of the groundwater microbiome, because throughout the last 2 decades at least, we see that there are some attempts to take into account the larger populations which are present in the environment. However, most of the studies were done using selected bacterial strain or selected bacterial groups. Microbiome or community level interaction studies were relatively rare. So we took these arsenic bearing sands from the alluvial aquifer and allow the groundwater microbiome to interact with this. Now this possibly allows us to elucidate the nature and extent of the geochemical alterations impacted or imparted by the groundwater microbiome. And for the purpose of today's lecture, I am going to highlight how this groundwater microbiome would actually show us that how the taxa recruitment, different organisms are recruited specifically to facilitate this interaction with the arsenic, and also the role of organic carbon in that. So what we did, we took the water, the arsenic contaminated groundwater, and we collected the entire pool of microorganisms. That is, we call it the microbiome is extracted, and the microbiome was allowed to interact with the aquifer sediment under laboratory anaerobic microcosms, and with no organic carbon added, middle one, and with organic carbon added. Now, this extraneous organic carbon that we have added is actually a mixture of lactate, benzoate, and pentadecane, because in our earlier studies, we have found that these aquifers are often having this low concentration of these aromatic and aliphatic hydrocarbons, and the microorganisms also have their potential to degrade different type of aromatic and aliphatic hydrocarbons. So following a 2 months incubation, what we observed that there is a significant change in the dissolved oxygen as well as the redox potential. So the dissolved oxygen showed that the conditions become more anaerobic, and the redox potential data suggested that it becomes truly an anoxic environment as the EH value goes to like close to a minus 60 millivolt or so, particularly when we have the organic carbon added into this microcosm. So this was coupled with significant bacterial growth, particularly if we look at the bacterial and archaeal growth through a gene transcript assay by extracting the RNA from these microcosms. You can see there is a two orders of magnitude increase in bacterial numbers from 10 to the power 6 to 10 to the power 8 in organic carbon added sediment incubation, whereas in no organic carbon added sediment also there has been some increase, but not very significant. The number of archaea, however, did not change very significantly under the similar conditions. Now, this change in the DO, change in the reduction potential and the microbial growth, all these things were found to be well connected to the release of iron, particularly as Fe2+ within the organic carbon added sample, although no organic carbon sample also showed some amount of iron release, but considering the presence of the scenario where we have organic carbon added, the concentration of Fe2 release was very significant. And with arsenic, the data was very interesting, because with respect to arsenic, we found that sediment with organic carbon led to a very, very high amount of arsenic release. And the total rise in arsenic concentration, if we consider, compared to only 65 microgram per liter into only sediment and microbiome, when we add organic carbon, it goes to like 400 plus microgram per liter, signifying the role of organic carbon in helping the microorganisms to possibly interact with the sediment and the minerals, and leading to the release of arsenic into the aqueous phase. Now, when we look into the speciation of arsenic, over a period of time, we observed that initially, particularly in organic carbon added sample, the arsenate and arsenite was these two levels were showing some interesting trend. Maybe in the initial time and also in the final time, we see that arsenate dominant, but somewhere during the intermittent period, it is the arsenite which is also detected, but in the elevated level, slightly higher level, which might indicate that arsenate, which is released initially from the mineral phases, are actually reduced to arsenic 3, and essentially arsenic 3 is more toxic than arsenic 5. So therefore, in a long run, the bacterial or the microbial strains or microbial species who are growing there, they are reducing this, or rather they are oxidizing this arsenic 3 to arsenic 5 again, and therefore the arsenic 5 concentration is again rising. Now, when we look at the solid phase of this sediment, we found that the iron oxide minerals, particularly oxides, along with the iron and arsenic, indicate significant changes. So I'm not going into detail of this, particularly the XRD data, which suggests the mineralogical changes, indicate that the highest release of arsenic is from the crystalline and amorphous, because we did a sequential extraction of arsenic from the different phases of the solid phase arsenic minerals, iron minerals. And we found there is a strong impact of the groundwater microbiome on arsenic iron oxide minerals like hematite, geothite, etc., as you can see with the XRD data. We also did the X-ray photoelectron spectroscopy to identify the relative abundance of this arsenic 5 and arsenic 3, which clearly confirmed that it is indeed the concentration, as you can see, that arsenate concentration from the control sediments towards organic carbon added sediment is significantly declining from 88% to 57%. We also noticed that the residual organic carbon, which was there with the sediment, is consumed by the microorganisms while they grow, they interact with the sediment. With the GC mass data confirm that the different level of hydrocarbon residues and organic compounds which are being consumed compared to the sediment alone, or the sediment with microbiome but no organic carbon. In presence of organic carbon, the residual hydrocarbons are all metabolized very significantly. Now, when we look at the microbiome, we found that there is a considerable change in the species abundance, like the operational taxonomic unit counts indicate that there is a significant change. But the more significant would be the taxonomic identity of the organisms. Now, compared to the groundwater microbiome, which is found to be mostly dominated by proteobacteria, and interestingly, Deinococcus was also there and few actinobacteria in the normal groundwater microbiome. But following incubation with sediment, the gamma and alpha proteobacterial abundance declined significantly compared to the normal groundwater microbiome. However, the abundance of bacteroidetes, which are anaerobic, known to be hydrocarbon and organic carbon metabolizing, but we did not add any organic carbon in this set, particularly only sediment and groundwater microbiome were allowed to interact. And finally, within the organic carbon amended set, it showed entirely different set of organisms, particularly the alpha proteobacterial members were found to be highly enriched or highly abundant, along with few members of firmicutes, actinobacteria, and some other members. So if we look at the data at the genus level, we will be able to find out that compared to the control sediment, which was having some members like Acinetobacter, Brevundimonas, Sideroxidans, etc., the sediment microbiome, when microbiome was allowed to interact with the sediment, the archaeal member, Thomarchaeotas, Sphingopsis, and some other members, Pseudomonas, etc., were enhanced. Particularly when the sediment microbiome, groundwater microbiome was amended with organic carbon, the presence of rhizobium along with E. coli, Pseudomonas were found to be very, very, very abundant. It was very interesting to note that the members of the rhizobiums were not generally found in the groundwater microbiome, at least at a significant level, neither when they interact with sediment alone, but they emerge as a major abundant, super abundant population when organic carbon is available. Now, we tried to find out the correlation with the observed geochemical or physicochemical parameters that we measured into the microcosms. With the major members, major operational taxonomic units, what we observed that the members like rhizobium and E. coli, they correlated very strongly, positively with arsenic, iron, and also with DO and ORP. Whereas some members like Sedimenibacterium, which was less known, and also the members like Geobacter, which are very well known with respect to their ability to interact with arsenic and iron perhaps, were not found to be that significantly important in this case, at least this correlation pattern indicates. Now, the quantitative assessment of the qPCR of different functional gene transcripts from the extracted RNA indicated the expression level of various arsenic homeostasis gene, as you can see, particularly the cytosolic arsenic reductase like ARSC, gone up to like 2 order of magnitude increase, and also the efflux, arsenate efflux pump, and also the cytosolic and the periplasmic arsenate reductase has also shown significant increase. The BSSA-like gene, which which is involved in the metabolism of the hydrocarbons, was found to be very highly expressed. And this could be attributed because of the presence of the hydrocarbons that we added through the organic carbon amendment. It is also found that the alpha proteobacterial member like Rhizobium that we found over there, Rhizobium belongs to alpha proteobacteria. The alpha proteobacterial members were indeed very active, metabolically very active, because again, a 2 order of magnitude in copy number only for alpha proteobacteria was noticed. Now the phylogeny of these major OTUs from both the sediment microbiome set as well as the organic carbon amended set were analyzed. So this is the phylogenetic analysis of the selected top OTUs from the groundwater microbiome sediment incubation. And this, the small black circle indicates the relative abundance of these. So the summary of this phylogenetic analysis is that the iron, manganese, and other metal-reducing geobacter and rhodonula type of organisms predominate when the groundwater microbiome is interacting with the sediment alone. Lactate, acetate, formate, fermenting, so fermentative microorganisms are also playing important role when the sediment is exposed to the microbiome, but of course, no additional organic carbon is there. Short chain fatty acids, C1 compounds, and different monoaromatic compounds utilizing organisms are prevalent, and ammonia oxidizing or nitrate reducing strains or species were also found to be abundant within this group of organisms which are possibly recruited by the microbiome when they are exposed with the iron oxide type of minerals. Now, the similar analysis done with the groundwater microbiome sediment organic carbon amended set indicates a something very interesting, that the Rhizobium type of members that we found. So most of the abundant OTUs are affiliated to Rhizobium. So a bunch of OTUs which are very abundant, up to 10% like that, you can see they are all Rhizobium. And what is more evident that these Rhizobium like OTUs are closely related to two other Rhizobium species previously reported from different arsenic contaminated and selenium metabolizing ability. So this was very interesting, that facultative anaerobic arsenic, iron, and selenium reducing and hydrocarbon utilizing Rhizobium species were surely there playing a very important role. Sugar and acid fermenting E. coli lactobacillus type organisms and polymer and hydrocarbon utilizing arsenic nitrate reducing Pseudomonas, Thoira, etc. were also found to be the major populations when the microbiome was supplied with organic carbon. Now, we have also isolated a number of strains in order to validate that indeed these Rhizobium type of members, they play a role in arsenic mobilization or in interaction with the arsenic bearing sediment. So we have isolated a couple of Rhizobium strain from this organic carbon amended sediment. So I am just highlighting that part of the data. And what we see that these Rhizobium isolates, so these are all Rhizobium, different strains of Rhizobiums isolated from the organic carbon amended set. And they show a very, all of them, they portray a very, very significant lineage with this, again, this Rhizobium arsenic reducens, which was previously isolated from arsenic contaminated groundwater, and it is capable of utilizing hydrocarbon and as well as reducing arsenic and iron, and also the selenium reducing Rhizobium strain. It is also interesting that these isolated bacterial strains showed a very good agreement, phylogenetic agreement with the OTUs, Rhizobium related OTUs, which we detected through the cultivation independent approach. Thereby, we were able to actually claim that we are now cultivating those organisms which actually predominate the real the community when they are allowed to interact with the sediment in presence of organic carbon. Now, these Rhizobium members were further studied for their abilities to utilize or reduce iron and arsenic, both as from the solid as well as the soluble phases. And it was found that they are both capable of utilizing the soluble as well as the solid phase arsenic and iron. And they accumulate those arsenic somewhere around the cells, as the ACM EDX data confirms the relative abundance of arsenic into the cell mass which is generated. So particularly the rhizobium species, those are tested, found to have the ability to almost like 80% reduction of the soluble Fe3 and AS5 and 40% reduction of the solid phase arsenate and iron, particularly when we provide the organic carbon as a source, carbon and energy source in the microcosm. So based on this study, we conclude that the groundwater microbiome of arsenic contaminated site are capable of interacting with the arsenic bearing aquifer sediment through specific engagements of microbial species. So there could be hundreds and thousands of species present in the microbiome, but it's not true that all the species are equally recruited by the community. So it's a community's collective decision possibly that they are recruiting few taxa when only sediment is available, or when along with the sediment we have the organic carbon available. The species recruitment was geochemically constrained. Particularly, organic carbon has a very important role in impacting the microbe sediment interaction. This surely validates the existing knowledge that organic carbon is playing a very important role, and it is easy to understand because it provides the thermodynamic resources, energy, the electrons, the carbons. Everything is going to come from this organic carbon. It is also found that the role of multipartite microbial processes, including the anaerobic utilization of hydrocarbon, which is found to be very unique in this case, because in other studies, we do not see much of the reports that hydrocarbons are being utilized during this arsenic dissimilatory reduction or arsenic mobilization processes. Microbial cytosolic arsenic reduction, iron transport, along with metal bioreduction and bioweathering of the clay minerals were also observed. Now, we also demonstrate the importance of the chemolithotrophic and heterotrophic members separately, perhaps, because during the no organic carbon amended condition, it is mostly the chemolithotrophic organisms who are playing important roles, whereas during the presence of organic carbon, it is interesting to note the role of rhizobium, which is perhaps not so much explored with respect to its potential in helping the release of arsenic, along with E. coli and Bacillus. Bacillus members are already known to play a role in arsenic transformation. Geobacter, another very well-known species or member, is also found. And it denotes that the anaerobic hydrocarbon metabolism coupled to arsenic reduction by both cytosolic and respiratory reductase-based activities dominated the rhizobium and Bacillus members. And this part of our work is recently published in the Journal of Hazardous Materials. And with this, I end my talk. And I acknowledge my funders, the Department of Biotechnology, Government of India, IIT Kharagpur, CSIR, Government of India, Department of Science and Technology, Government of India, past and present students, and all the collaborators and the FAMES for organizing this meeting and giving me the opportunity to present our work to you. With this,
thank you, Professor Sir. Thank you, Professor Sir, for a wonderful presentation. And this is really loaded with information. Ben, do we have time for one or two quick questions?
Absolutely, absolutely.
Okay, Professor, I might have missed, what is the organic carbon you are adding in the experimental setups? You are muted. Yeah.
We are adding a mixture of lactate. We are adding pentadecan, and we are also adding some aromatic hydrocarbons. The aliphatic, aromatic, and these sugars are all there.
Okay. Okay. And do we have a study of, like, once the organics are exhausted, how the microbial community changes?
When we stopped the experiment, that is after 2 months, our GC mass data showed that organic carbon is nearly exhausted, though we are not claiming that we're not claiming that the organic carbon is absolutely exhausted. Okay, but that can be another interesting study, that given a chance that the organic carbon was initially available, then it was completely depleted, how the community behaves actually. So
yeah, I guess once the carbon get exhausted, and also with the time the iron and arsenic will also get removed, like maybe co-precipitated and removed. then because the toxic levels are reduced, maybe the other communities will pop up. Like, as you say, the taxa recruitment changes with the structure, with the microenvironment. So
that is absolutely expected.
So I'm curious, you have seen any methanogenesis in your study?
Methanogenesis, no, but the presence of Thomarchiota in no organic carbon added condition.
Okay.
That we have noted that during with no external organic carbon added, Thomarchiota members were there. So possibly some amount of methane may be generated, but one thing we have observed that C1 compound utilizing organisms are very abundant. So whatever low concentration of methane is generated, and you might be aware that recently the ONGC, they have found oils, petroleum oil reservoirs, and very soon the exploration of oils are going to be started in this region. So it is expected that methane would be there somewhere. And this methane may not be as a methane gas, but methane is transformed into alcohols. So it's basically the methanols and other C1 compound, methylated compounds. So we see a lot of bacterial strains who are capable of utilizing C1 compounds.
Right. Okay. Okay. That's pretty interesting. Okay. Thank you so much, Professor Sar. Ben, do we have any questions from the audience?
No, I didn't get any questions but yeah. So yeah, with that, I mean, I'd like to thank you. Well, thank all three of you. I see that Saugata is also still here. So thank you very much for your contribution and for a very, very interesting session. And thank you for persevering with the technology. You know, it's not easy kind of in these kind of high pressure meetings like this, but I appreciate you kind of working through, persevering. We actually got to hear a lot of really, really nice information. So yeah, thank you very much. And I'll say goodbye to you now, and we'll move on to the next.